All right, good afternoon, everyone. So here we are virtually and extremely pleased to see the roster all filled up here. So we shall go ahead and start our first session. It is June the 1st, 2021, just about 2:18 here. So we'll basically start with some brief introduction and also the course setup. So first of all, as hopefully you guys have been keeping up with the emails that I've been sending out over the past month or so. Uh, just as a brief background on myself. Um, so basically by training, I am a doctor of medicine um, and not a teacher really. That's not at least how I started out. So basically I finished med school back in 1999 and then took my USMLE exam, which again stands for the United States Medical Licensing Examination, which had 3 steps back in the early 2000s, step 1, step 2, and then CSA, which basically stands for Clinical Skills Assessment. So it was a rather lengthy, painstaking, expensive process that I went through. And eventually I ended up securing a medical residency in internal medicine at a downtown, very busy downtown Chicago hospital, where I did basically my 2 years of internship, first year of residency in internal medicine back in 2003, 2004. And at that point, somehow the crazy idea struck me that I would be better off teaching this stuff. Okay. Basically, it was more of a personality mismatch, work uh, life balance, whatever you want to call that. At any rate, I made that decision to switch to teaching, and I've been teaching ever since, and have been extremely fulfilled in my teaching career. Um, so as of now, in addition to being an adjunct professor of human anatomy and physiology and occasionally college biology at TCC Northwest, I also have a full-time teaching job as a high school pre-AP and AP biology teacher at a charter school down in Euless. So that keeps me plenty occupied during my weekdays. And I do weekend classes for TCC and occasionally evening classes as well. And that's what it seems to be what I'll be doing come this fall as well. All right, so with those words of basic introduction, let me take you here to our course uh, and talk about some of the things that pertain to that. And I've been sending out a number of emails, right? Hopefully you've been keeping up with checking those. And so that's something that you will continue to do over the course of the semester. All right, so on your Blackboard page, you're gonna see something like this, right? So here's our course, BNP1. And so as I'd mentioned in my emails, you are going to look for this option here, lessons in the left-hand column. So I'm gonna go ahead and click on there. And when you click on lessons, you see these number of documents that have been uploaded. All right, so here you have all of your PowerPoints that we will be covering over the semester, right? All 16 chapters are here. And then you also have those three, CID, which basically stands for Course Information Document, the LID, which is your lab information document, and then frequently asked questions, all right? So let's quickly take a look at each in turn and try to understand what this course looks like, what are the expectations, what you guys will be doing, and what are we aiming for at the end. All right, so first if you click on CID, which is your course information document, and have them all opened up here. So let's see. All right, there we go. This is what your CID, course information document, looks like. All right, so all of that important information, my name, my physical office address, which has been quite redundant over this past year or so, year and a half, thanks to the pandemic that we've been facing. And thankfully, we are seeing a downward trend there now. So we are all set for in-person learning come this fall. The most important piece of information that you have here is my email address right here. So please make sure you use this to get in touch with me. All right. Also, when typing this out, do not rely on the web advisor email that is given, because as I found out over the past semesters, they had messed up uh, my name spelling, and so none of the emails were getting to me. So please make sure you put the email exactly as it is here, right? So the letters in order. Uh, it's not unusual for me to be called Dr. Fraud Khan, and so I'm not a fraud. At least I like to think so. So it's Fuad, F-U-A-D. So please make sure you get the letters in order, .con, Khan, at tccd.edu. You can also use my Gmail account. So if you add a little my here, all right, so like my.tccd.edu, that's fine as well. In fact, that is the email account that I use to get in touch with y'all, and also for sending out the recorded lectures and the notes. Uh, and I will be talking about all of that here briefly. My office hours, okay? And then again, this being all virtual, I'm potentially always there. I have this online presence, so you can email me at any time, ask questions, feedback, suggestions, whatever you have. So here is the course description, what we will be covering. Of course, the anatomical and physiological sciences, as you can see here, including labs, online labs in this case, course schedule, course focus, uh, transfer considerations, okay? And then here is the required text and references. So this is something that a lot of students have questions about, right? So what we are looking at here is the textbook from which the PowerPoints are derived. So as I'd mentioned in my emails, if you have the textbook, that is great, wonderful. That is your firsthand source of reference right there, okay? You don't necessarily need a new textbook either. You might have an older edition, which is just as good. However, as I'd mentioned, if you purchase a new textbook, you do get, or you should get, a Connect access code, the Connect website that I will be mentioning here shortly as well. So that should come with a new purchase. If for whatever reason, as one of the students just notified me, she apparently did not get an access code, please get in touch with the bookseller and ask them about it, because that should be the case. You should be getting a free access code if you purchase a new textbook. In any case, all of the PowerPoints that we will be going over are based on this textbook, okay? So, and those PowerPoints have been uploaded, as we just saw, on our Blackboard page, okay? So as it says, it is not advisable to attempt the course without the use of a textbook. So yes, it's definitely an advantage to have it, right? It's not technically mandatory, but it's going to help a whole lot, because that is your body of reference for all the information, okay? All right, so then, in addition, as I'd mentioned, you also will need the Connect website, and you get to that Connect website using the link that I sent out in my emails earlier. So it's a paid subscription. I think they have a 2-week grace period or something that you can have free access, but then from that point onwards, you will need paid access to take your lecture quizzes there, okay? All of the lecture quizzes are posted on there, and I also detailed what are some of the options as to how to turn in those quizzes, right? So you can do them all in one stretch, or wait and follow this schedule, the timetable, and take them in order, which is preferable, or you can just do everything all at the end. As long as I have all of those 7 quizzes turned in by the deadline, you should be good, and I should be able to grade those, okay? All right, student contributions, at least 9 hours per week, comes about, what, an hour and a half on a daily basis. Some of you would have more time than that to devote, which is great. Some of you would be able to do just as well with lesser hours put in than that. So everyone should be able to come up with their own pace, their own schedule. One thing I highly recommend is forming study groups with your classmates, all right? Bouncing questions off of each other, look for interactive websites, YouTube, Wikipedia. I have no objection to using any of those resources. They should all come in handy for preparing for the course, all right? The more sources, the better, the clearer your concepts as well. Students will attend class, in this case, recorded lectures. So if you have any unforeseen circumstances where you have to take, well, you still have recorded lectures here, but at least let me know if you will not be able to, or were not able to go over the information for whatever reason. So at the very least, I will have some idea as to what's going on with you there. All right, so your quizzes are all online, lecture quizzes that is, and they will be uh, available at the Connect, they are available at the Connect website as we speak. So something to keep in mind, okay? And so here is your point distribution. So this is where the real meat of this whole course is for a lot of students, all right? So what that point distribution looks like is you have 4 exams here, 4 online lecture exams. Uh, all of these exams will be posted on your Connect website as well. So that's why you need access there. All your uh, lecture quizzes and your lecture exams and your lab practicals will also be, and your lab activities as well. Some of you already turned in your first day's lab activity, which had to do with lab safety and so on. So all of that is on the Connect website. So these lecture exams are 4 in number, and each one is worth 100 points. And so how those exams work basically is you will have 10 questions, all right? There will be a total of 30 questions, each worth 4 points, okay? Each worth 4 points. So you might be wondering if you have 30 questions and 4 points each, so that's what, 120 points, and it says 100 points. So what that tells you is 20 points are kind of extra credit for you. And so how this whole thing is structured is the first 10 questions on those lecture exams will be based on a study guide that I will send out in advance of each exam, right? The rest of the 20 questions will basically be based strictly on these lectures that I'm going to share with you. Okay, the recordings, so you have to watch the entire length of the recording, uh, take your own notes. I will be taking notes here as well, and I will be sending out pictures of those notes. And that is extremely important because all of these 20 questions are based off of that extra information. So there's a lot of clinical scenarios, clinical pearls of wisdom, if you will, personal stories and anecdotes that I throw in there. All right. So that's not something that you can Google and come up with an answer for those. All right. So I'm not that famous till this point, at least to my knowledge, that my entire life is chronicled on Wikipedia or something. So you have to be basically listening to the lectures and paying attention to the notes that I send out in order to be able to answer those questions, okay? So basically that's what it is. You will get a study guide, but that will only refer to the first 10 questions. The rest of the 20 questions are based on the lectures, the recordings that I'm going to be sharing with you. So 100 points in all, 120 points is what you have, right? But as soon as you make a score of 100 points, any extra score that you might have attained on there, that is irrelevant. So you'll get 100 out of 100. That's it. All right. It's not like a credit card accumulating system, unfortunately. So those uh, that those marks, that credit doesn't roll over to your next exam. The next exam will have its own standalone set of questions. All right. So that's 400 points for your lecture exams. Then we have 7 quizzes, as you can see here, and that's an E grade, meaning these are all, none of that, this is paper pencil, of course. These lecture quizzes are on your Connect website. And each of these quizzes is worth 15 points. You have 15 multiple choice type questions there, okay? Each worth 1 point. So taken together, that's 105 points for your lecture quizzes, okay? And finally, we also have something called case studies. Not finally, we have something else in terms of the comprehensive exam as well. So the case studies are basically these research papers. Think of them like those, all right? And I will be sending out guidelines for how to do those case studies as well. So basically, you will be choosing a disease entity from any of the body systems, like nervous system, cardiovascular system, endocrine system, whatever, and basically preparing a full presentation on that based on the guidelines that I will be sending out. And that would be worth 50 points for y'all, okay? And then finally, you have the big comprehensive final exam, okay? Sounds pretty scary. Hopefully it's not going to be because I will be sending out a study guide for that as well. So the comprehensive lab exam, or final exam, sorry, is 50 questions long, okay? Not the usual 30 questions that you have for your other lecture exams. This is gonna be 50 questions. Out of those 50 multiple choice type questions, all posted on Connect website, of course, You will have the first 25 questions based on a study guide that I will send out again. The last 25 questions are based off of all of the lectures that you will be reviewing at the end of the course, okay? So all of that information and notes are fair game at that point. So 50 questions and each is worth 3 points. So that's 145 points for your comprehensive final lecture exam, okay? All right, so then the rest of the 300 points come from your labs. And I'm going to talk about your lab portion separately here, okay? So taken together, you have a total of 1,000 points at your disposal. So don't think about passing lecture and lab portions separately. There's no such thing. It's a cumulative thing. The better you do on each and every assignment, the higher your grade at the end. Think of it like that. So don't try to break it down into, okay, this is lab, this is lecture, this is more important, this is less. No, every single thing that you do is going to count for you, okay? And you have those extra 20 points available each lecture exam, as we just mentioned. Okay, so 1,000 points total. In order to get a numerical grade of A, you have to score at least 895 points and above. For a B, at least 795 points and up to 894 points. For a C, from 695 to 794 points. A D is from 595 points to 694, and a total score of less than 594 points. That's not a passing grade as we see here. Now, based on your performance and how many brownie points you score with me, just kidding. You can score your brownie points if you are sending me a lot of informative emails, articles, medical stuff that you might find interesting, case studies, and sharing them with me. I enjoy going over all of that. So in any case, if I see diligent effort from you, and let's say you fall about 5 points less than what is needed to make it to the next higher numerical grade, right? Let's say you end up with an 890, and you need 5 points to make it to 895 and higher to get an A. Usually I will grant you those points. All right, again, looking at your effort, your presence, and all of that, okay? Rare circumstances, even if you're at an 889, right? 6 points shy of making it to the next higher grade, based on, again, your performance, I would consider bumping you up to that next grade, something to keep in mind. All right, so here, what we are seeing is our timetable and the schedule, okay? So quite important. So this tells you exactly what topics we will be covering each day, all right? So, all right, there, the date, and then the content that will be covered. So as you can see today, we are going over course introduction that we're talking about right now. And then I will be covering chapter 1, the sciences of anatomy and physiology, okay? For tomorrow, we have chapter 2, atoms, ions, and molecules, so on and so forth. And as you can see, that are quizzes. These are the recommended dates. So for example, if you are sticking to the schedule, which is recommended, preferable, your Connect quiz number 1 should be turned in by this Thursday, the 3rd of June, okay? You would be in good shape to turn that in because we would have covered all of these topics that pertain and apply to this quiz here. And then as you can see, next week on Tuesday, the 8th of June is when we have our first exam, online exam. And I will be sending out a study guide over the weekend so that you guys can go ahead and start preparing for that exam as well. So take a look at the schedule. This is what we're going to be sticking to, and that gives you a good idea of what we'll be covering, when those exams are, what to expect, okay? And usually I will give you an entire day almost to take that exam, all right? So the exam would be an hour long, 30 questions, and you'll get an hour, 60 minutes for those 30 questions, and you'll have a day to take that exam at any time, all right? You cannot exit. Once you start the test or the exam, you cannot just like suspend it or exit and come back or something like that, right? You have to take it in one sitting. Once the time starts clicking, you will have 1 hour, 60 minutes to finish those questions, but you can space out that hour according to your schedule, home routine, whatever, on that day, okay? And I will be mentioning all of this in the study guide that I will be sending out. All right, so here is um, your timetable, your schedule, your syllabus, whatever you want to call that. And here is your comprehensive final exam. That would be when? That would be on the last day of June. So that's not a 7, that should be a 6, right there. That's 6th, June the 30th. So, well, I'm going to save it at my end. But yeah, you see what that is. Okay, so that is the course information document, everything you need to know there. So I'm going to go ahead and close it here. Now, let's go ahead and take a look at the lab information document. All right, LID here. So much of the same information, my name, course number, my email address, that's important. Again, do not mess up my name spelling. No fraud cons or Dr. Frauds there, hopefully. And you can, again, you can type a my.tccd in front of that as well, or just tccd. Both of those inboxes are regularly checked by me. Office hours, which do not really apply now. I'm in my office literally 24/7 virtually. Okay, so here's the question. A lot of you were uh, asking about, okay, so what do we really need for this course? So I mentioned two things. You need access to your Connect website using the link that I sent out. And this is a paid subscription again. This is not something that I came up with because I wanted to give extra money to the Connect people. This is something which is mandated by the district. So we all go through that, all right? So Connect website access for taking all of your exams and all the things that I mentioned. And you also need either a paper copy or an e-version or electronic version. I have no preference. Both are equally fine. Uh, of this lab manual, Exploring Human Anatomy and Physiology in the Lab by Aaron C. Emmerman, the 3rd edition. You can have like a secondhand copy bought dirt cheap off of Amazon, doesn't matter, all right? Or the electronic version, the ebook, whichever, they're just all fine. Um, but you need some kind of access to that lab manual so that you can work on the lab activities and turn those in. When I say turn those in, how will you be turning those in? So you handwrite your answers for pre-labs or whatever. As an example, take a selfie with your answers. Take a picture of your answers and your own picture with it and email me. Simple as that. All right. And sometimes you will have to email me repeatedly because the pre-labs are a little longer, whatever. That's fine. I will basically grade your work and I will put the grade in. Okay. Uh, if you're doing it electronically, again, you can take a selfie with your work, the screen showing your work done on the lab manual, okay? Or you can even share that screen with me, send me a screenshot or screen share with me, that's fine too, as long as I can see that you worked on those labs, okay? Again, do not worry too much about getting everything technically correct the first time around and turning it in. I'm looking for effort. As long as you're sticking to the schedule and turning in whatever you can to the best of your ability, you should be in good shape, okay? So again, this all being virtual, I'm not looking for 100% technically correct answers or work or whatever. I want to see effort on your part, that you are doing your part and uh, staying in touch with me, letting me know how it's going. If you have any questions, feedback, you have my email address, please let me know, all right? So this is a two-way street. I will be in contact with you, you all sending out communication and you do the same at your end, okay? So do not get stuck on getting everything done perfectly well the first time around. Email me if you have questions, all right? So here is the point distribution again for your labs, the lab section, of course. So what you see here are 4 lab exams, and they are, okay, so not 70 points, but they should be 50 points each, okay? So 50 points each, I'm gonna make that correction here, and you will see that correction at your end as well, okay? Now, all of these lab exams are going to be posted on your Connect website as well, okay? And I will send a study guide, what that lab exam looks like. So typically you'll see some images, pictures there, and multiple choice type, fill in the blank, or true or false type questions that go with those pictures, okay? And so as long as you're doing your labs on time and studying the material and going over it and have some working knowledge of those topics, you should be able to get done with those lab exams without much trouble, okay? Uh, 50 points each. And so how many questions that is, I will be sharing that information well in advance of your first lab practical, okay? And we'll have 4 of those all on the Connect website. Okay, so 4 lab exams, 50 points each. So that should be 200 points, right? All right. Then it says uh, 6 quizzes at 15 points each. All right. So what are these 6 lab quizzes? Okay. So these are not separate quizzes. This is nothing that you will be working on separately on Connect or paper pencil or anything. All these is the daily work in labs that you will be turning in, and I already sent out what that work is, right? Some of you already finished your lab safety virtual labs on Connect. That was it. And then I also sent out today earlier an email which talked about what is due by tomorrow midnight, right? And that has to do with enzymes in chemistry and microscopy and stuff. So that is all you have to do. As long as you're turning that in, I will be taking those activities at random without telling anyone which activities I'm going to be grading. And I will be taking your lab quiz grades, these 15 points from those, okay, at random again. So that's all you have to do when it says 6 quizzes. Those are simply the lab work that I will be sending out every day, work day that is, and the work that you will be turning in electronically to me. Those will basically count as your labs, all right? So the exam format, as you can see here for your labs, you will be shown images, models, microscope slides, and or specimens, right? All in on your Connect website. And then, all right, it says in a handwritten format. That's redundant. There is no handwritten format. It's all electronic, okay? So yeah, a few things that I will have to go back and fix here. Okay, quizzes, makeups, content. Okay, here. This is important. Again, this is your lab schedule. So these are the activities that will be done on a daily basis. Here are the dates, what we will be working on and you will be turning in, right? One of the students asked me about the rat dissection. Unfortunately, as much as I love them, we won't be able to, and I don't know if you guys love them just as much as I do. Some of you might, some of you might not, but we won't have that opportunity to do it in person, at least not for the summer semester. But we will have something, an electronic equivalent of that, okay? All right, so here are your exam dates. Practical exam number 1 on the 14th of June. So that is in bold red color, as you can see, okay? So there you go. This is your schedule. Now, as you can see, there is no comprehensive lab exam, okay? That comprehensive exam is only for your lecture. For labs, you will just work, take these 4 lab practical exams. There would be no composite comprehensive lab grand practical or something of that sort at the end, all right? Just in case you guys are wondering. Okay, so that is pretty much it as far as your lab information is concerned. So I'm going to close this one as well. Finally, here are some of the frequently asked questions, okay? And I compiled this list based on the most frequently asked questions that I get semester after semester from students, okay? Such as, what is this class all about? How will it help me in my future career? Well, this class is obviously all about an introduction to anatomy and physiology, but even so, based on my own medical background as an internal medicine doctor, uh, this could also be considered a kind of like a pathophysiological class. A lot of pathology is what I like to discuss. So disease processes, right? Anatomy and physiology is, of course, the normal functioning of the human body. Pathology is the pathological or diseased functioning of the human body. So oftentimes I will backtrack rather than starting with the normal function. I will throw out a clinical scenario at you that is about a patient who obviously is not doing so well. And from that, we will go back to the normal function, okay? So you can expect a lot of pathology thrown in there as well. But basically, we talk about disease conditions, health maintenance. As we see, it will help you in your nursing, allied health, respiratory or surgical technician career, whatever. It will help you increase your general knowledge of the functions, the functioning and the structure of the human body in general, okay, and how to maintain your overall health if you never go into a medical career in the future. Even in that situation, it's going to be quite helpful. What am I expected to do? Well, pay attention to all the emails, listen to the recorded lectures and the notes that I'm going to share with you all. Take your own notes here, right? Maintain regular contact with your instructor, put in the necessary time and effort in order to be successful in this class, all right? What is the schedule like? We just looked at what the schedule is like. It's posted both at my TCC. It's also at WebAdvisor. You can always look at it there as well. Grading criteria, I already talked about that. It's also posted on there. What does the professor really teach and emphasize? So again, based on my own background as a doctor of medicine, whether I like it or not, or whether you like it or not, usually students like it. So a lot of pathology, disease processes, and clinical medicine. So clinical techniques, clinical examination techniques, history taking, those kinds of things, and clinical applications is what we will be talking about over the course of the semester. Is there any extra credit available? I talked about that as well, right? So you have 20 points over and above the usual 100-point exam that you have. And I talked about how that is structured as well. Any curve done on the test? Well, that is the curve. The curve is you have 20 extra points every lecture exam available. How do you know the content of the forthcoming exam? I am going to send out a study guide for each of them, as you can see here, okay? If I run late for whatever reason, because I will be busy this summer, I'm also teaching summer school at the charter school where I teach AP biology. Please do not hesitate to send me an email and remind me, hey, Dr. Khan, we have a lecture exam number 5, or well, there is no lecture exam 5, or lab exam 4 or something coming up. So if you don't mind, please send us the study guide, okay? What are the professor's favorite topics and strengths? Psychiatry, which again is the study of mental disorders, okay? Also gynecology, the female reproductive system and disorders, urology, kidneys, male reproductive system and disorders, tropical medicine, immunology, and neurology, okay? So these are, and I misspelled neurology there. Some of my strengths and kind of favorite topics. And I came up with this list based on my USMLE scores. When I took that medical licensing examination, it actually gives you a performance bar on how you did on each of those topics. So these were some of the areas where I scored highly. So this is where I've derived this information from. What if I miss some classes, which means you do not basically review the lectures? Will it affect my grades? Of course it will, because as I mentioned, 20 questions out of the 30 would be based off of those lectures, the recorded lectures that you guys are going to receive, and also the notes that I'm going to share with you, okay? So make every attempt to be diligently present there, okay? So how to bring up your score? Study seriously and in timely fashion. Take notes in lectures, pay attention to study guide, and most important of all, good attendance, which means, again, reviewing all of the material that I send out. Some good study habits and strategies, flashcards, very helpful, interactive websites, mnemonics, acronyms, most importantly, constant revision and repetition. Okay, this is all I did during my medical career. Repetition, do it over and over again. Ask questions, right? Form study groups, and study with each other. Now you have online resources. I didn't have the type of online access that people have now, right? So take practice exams. Just pull up practice free A&P type 1 MCQ type exams and take them, and then go back and fix your mistakes, all right? The more multiple choice type questions that you take, the higher your score in general, all right? So that is some good advice that I found useful even for myself personally. My qualifications, I'm an MD, doctor of internal medicine, and I have a cumulative five-year experience of hospital work, all right, as a first-year resident, as an intern, and also as a medical student when I did all of those clinical rotations. Can I record or tape your lectures? Well, this is redundant. I am recording those lectures for you. Can you upload those if you want? Yes, you can. What if you still have questions? Email me here, okay? And I've shared this information with you as well before. So there we have the frequently asked questions and their answers, all right? Okay, so let's go ahead and sticking again to our schedule, we are going to start with the first chapter here, and that is the sciences of anatomy and physiology, okay? All right. Okay, so here we go. So what are we even talking about when we say anatomy and physiology, okay? Let's break down these terms. Anatomy, anatomy. What does that mean, okay? It's a composite of two different terms. So ana here means across something, okay? Ana means across. And so then what is tomi? To cut. So what does that mean? To cut across. That's what anatomy is, because of course the knowledge of the internal structures of any living thing come from cutting through that structure and analyzing that structure, right? So that's basically what the science of anatomy is all about. Cross-cutting and looking into and studying the organs and the cells and the tissues and the body structure of an organism, okay? And the person who could be credited with providing our first-hand knowledge and some of the very first laying down the foundations of human anatomy is a Roman physician by the name of Vesalius, okay? And Vesalius was the person who also was passionate enough about human anatomy to the point where, well, during those Roman times, it was considered desecration to basically dig up dead bodies and, you know, or cut into the human body at any time. So what he was forced to do, what he resorted to doing, was paying people who under the cover of darkness will basically go and dig out bodies, all right? So grave diggers, if you will, or grave robbers would be a more appropriate term, okay? But it was not for any nefarious purposes, nothing else than performing kind of like a post-mortem exam, an autopsy, and looking into the human systems, all right? So we can thank him for much of the modern anatomical knowledge that we have. All right, physiology. So physiology, the word logi comes from the Greek word logos, right? Or logos, which means study of, okay? Study of. And physio has to do with the function of how something works. So physiology is the study of the function of the human body, okay? Now, and it says form and function are interrelated. Very important, okay? Anatomy and physiology cannot be studied in isolation. I mean, these are two different disciplines, sure enough, but they go hand in hand. You cannot totally and completely and really understand one without knowing what the other is talking about, okay? And I'll give you an example here. So let's talk about the structure of the human foot, okay? Now, and why I picked the human foot is because this is one of the most unique biological structures and organs in existence, all right? No other creature has a perfectly bipedal gait, bipedal, standing upright on 2 feet, 2 legs, and walking. Only humans as a species do that, all right? All other creatures like apes or bears or whatever you're thinking of, they're primarily knuckle walkers. They walk on all four, but they have this ability to sometimes rise upright for short distances for spotting their prey or sniffing their prey out, or danger or whatever, but then they go back to that, right? And birds technically don't walk. They hop or just walk for short distances. They're flyers, of course, right? Humans are the only truly bipedal species. So if you look at the human foot, again, you see that we have 5 toes, all right, and they're all in the same plane. And this is something which distinguishes the human foot from our most closely related animal cousins in the animal kingdom, the great apes, gorillas, orangutans, chimpanzees, bonobos, so on and so forth. If you look at their feet, they also have 5 toes, but their big toe is opposable. Their big toe, it's like our hands. Their big toe is opposable because they use it to grasp onto objects like branches and twigs and even pick up objects with that. Humans can't do that because our foot is perfectly evolved for one thing: walking, bipedal walking. So our big toe is in the same plane. It's not an opposable toe as compared to the other ones. Okay. So if you look at the human foot, what would be the anatomy? 5 toes, the big toe in the same plane as the little toes, all there, right? We have arches in the feet, right? We have the longitudinal arches and the transverse arches that we will talk about later on during the course of the semester. And so these arches are made by the muscle tone and the ligaments of the foot themselves, all right? So an anatomist is simply studying this, but the anatomist is also a physiologist. So what is he thinking as he's looking at the foot? Wow, well, this creature must walk on 2 feet, because you can see that the big toe is in perfect alignment in the same plane as the little toes. This is perfectly suited for a bipedal walk on 2 feet, right? And also the foot is elongated as compared to the apes, as an example, because they have like more of a circular foot, right? Which also distributes the weight for bipedal upright walking perfectly, right? Um, and you are also noticing the arches, the longitudinal and the transverse arches in the foot, which also corroborates your assumption, your conclusion that this is a bipedal walking creature. Okay, so let me put this human foot as an example of interrelation of anatomy and physiology, right? So there you go. You cannot truly and really study the anatomy of a structure without understanding or inferring the physiology, or and vice versa. Okay, so these are interrelated sciences, both anatomy and physiology, as we amply saw here in this example. All right, let's see. Now, the next thing that we are looking at here is something called the scientific method. And scientific method is what defines all sciences. If you claim a discipline to be scientific, it has to follow the scientific method. And what is the scientific method? There's 5 steps to it. If you've ever participated in your school science fairs or college science fairs or whatever, you're well familiar with with what the scientific method and what the 5 steps are. Excuse me. So, number 1, you start with a question or an observation, right? For example, um, why does the human heart rate or pulse differ from person to person or from time to time? Okay, that's a valid observation and a question. So, excuse me, what do you do if you have an observation or a question? Uh, you come up with a hypothesis, and a hypothesis is an educated guess. So you might hypothesize that the human heart rate or pulse rate differs because of different types of activities that one is engaging in, right? Or um, the pulse or the heart rate might differ between 2 people based on their body mass. So someone who's bigger, heavier will have a faster, higher heart rate as compared to someone who's more athletic. Okay, so these are all educated guesses or hypotheses. But how do you know if your hypothesis is correct? Then you go on to the 3rd step, which is setting up an experiment. So how do you set up an experiment? Well, you take a person and you have them check their pulse rate, their radial pulse here, right, in the wrist, while they're sitting and relaxed, and then while they are lying down and relaxed, and when they're standing up, and then when they're like doing jumping jacks or something. Okay, and so you compare uh, in the same person, right? So that would be one example of setting up an experiment. And you take down, and so what do you do from all of this experimental setup? You get some data. So you make a data table, activity, lying down, sitting up, standing up, exercising, and then you write down the pulse rate. All right. So that would be your data collection. That's the fifth step. And finally, you get to the conclusion or the result, which is the fifth step. All right. And you say, yes, based on my data results, I conclude that your heart rate or your pulse depends on the type of activity or the emotional state that you are in. There you go. That is the scientific method. Observation or question, hypothesis, experiment, data collection, conclusion. Simple. And this doesn't have to be like some fancy science fair project that you're working on. You do the scientific method when you prepare a dish, as an example, right? So if you're just learning to cook or cook a new dish, a new recipe at any rate, uh, you experiment. You have a question. Okay, what if I add more salt or more spices to this or something? What's going to happen? So then you hypothesize this will make it more tasty or more hot or something like that, right? And then you actually cook the dish, and then you feed it to different people. And if they survive your experiment, right, or you survive your own experiment, which is your data collection, people are alive and healthy and happy, and they're praising your cooking skills, your culinary expertise, then you come up with the conclusion that, all right, so whatever I hypothesized, it worked here, right? So that's a very rough example of a scientific method application here as well, right? So cooking as example of the scientific method. Okay. So let's start with some subdisciplines, some different branches of both anatomy and physiology. So the first one we are looking at here is something called microscopic anatomy. So it's self-apparent as to what that is all about. Looking at structures which are too minute, too tiny to be seen under the regular, with your naked eye. So you have to put them under the microscope, like cells and tissues and so on, right? That's microscopic anatomy. If you're studying individual cells, that's called cytology. Cyte, C-Y-T-E, means cells. Logy, again, is the study of. Cytology is the study of individual cells, okay? If you're looking at tissues like fat tissue, which is called adipose tissue, or muscle tissue or nervous tissue, that is called histology. So by the way, anatomy and physiology are the two basic medical sciences that are taught during the first 2 years of med school. And then anatomy has two subdisciplines which are taught along with gross anatomy, of course, and those would be histology, number one, and embryology. Embryology, the study of embryos, embryonic development, okay? So here you have gross anatomy, and gross anatomy, of course, is the anatomical structures that you can observe with your naked eye without the aid of a microscope. So what are the branches of gross anatomy then? Systemic anatomy, okay? What is that all about? Systems anatomy, all right? For example, if you're studying the cardiovascular system, you are going to be looking at what? The cardiovascular structures. Cardio means heart. It's Latin for heart. Vascular is Latin for vessels, blood vessels. So heart and the blood vessels, you will be studying the heart, the blood vessels, which include the arteries, the veins, and the capillaries, and also blood itself. So that's a system, systemic anatomy. Let's say you're studying the endocrine system. What would you be looking at now? All the glands in the human body, the pineal gland, the pituitary gland, thyroid gland, pancreas, thymus, all of those, right? So that is an example, yet another example of systemic anatomy. So you study systems. Then you have regional anatomy here. So what are we talking about here? You study regions of the human body. For example, and this is how I studied, they teach anatomy in med school. They teach it in terms of regional anatomy. For example, when I started out my medical studies, first year of med school, the very first body region that I was taught was the upper limb. I was taught the upper limb first in med school, right? So, but my friends, they were in different sections, class sections. So they started out with different regions. So one of my close friends was actually started out with the thorax, um, the thoracic region. All right. One of them started with the abdominal region, the abdomen. Yet another one was starting out with the lower limb. Okay. So different people and different regions, but that's what regional anatomy is all about. All right. And believe it or not, it took us 4 months, a full semester to just cover one body region. So, for example, for me, the upper limb took one full semester, 4 months, because I had to memorize every muscle, every bone, every joint, uh, every movement uh, of the upper limb, every nerve, every artery, every vein that supplied it, right? And not only an in-depth knowledge of all of that, but all of the pathologies, things that can go wrong, the diseased states of the upper limb as well, right? So that took 4 months, and even after that, you still have some rough idea of what all of that is, okay? And then you move on to the next body region. So lower limb, same thing, another semester. Then the head and neck region, another semester. An innumerable number of structures that are involved in each region. Okay, so that's how you are taught med school anatomy, regional anatomy, that is. Surface anatomy, something else that we were taught. What happens here is, so what they used to have during my med school years is they actually had paid models who will come and disrobe, strip in front of the class, right? Some of them might have been professional strippers, right? But what we were asked to do on these models were, and they had well-defined like muscular bodies in general, right? So you could literally see the muscles, six-pack and everything on them. So the professor would ask us, all right, so I would like you to mark out the left clavicle on this person, or the brachial artery on this person, or the triceps muscle on this person. So we had to go with a like a water marker or a chalk or something, and we literally had to draw the outline on this model. All right. So that is what surface anatomy is all about, right? Actually tracing out the underlying structures on the surface of an organism, right? So we make some notes here as well. Surface anatomy and uh, strippers in med school. All right, so then comes uh, comparative anatomy. What is that? Well, as the term itself suggests, comparing anatomy of one species with another. In most cases, we're talking about human anatomy compared with other great apes. So we are on the same line, right? Like orangutans, bonobos, chimpanzees, gorillas, of course. So close to related species when you compare, or pigs for that matter. So for example, in TCC labs, we were dissecting pigs or pig embryos as a substitute for human systems. That would be an example of comparative anatomy as well, right? And then embryology, and I mentioned that one before. Embryology is the study of embryos, embryonic development. And this is also a branch of anatomy that we were taught during the first 2 years of med school, embryology and histology, okay? So um, first 2 years of med school, uh, okay, anatomy, gross anatomy divided into subdisciplines of histology and embryology. Okay, so that's how we learned it. Okay, pathologic anatomy, what does that tell you? Not normal anatomy, but anatomy that has been pathologized. Some disease process has involved here. For example, if you look at the lung of a healthy, non-smoking, exercise conscious person um, at an autopsy, or even before autopsy, if you take a biopsy of the lung or something, and then you study the lung of someone who was a 5 packs of cigarettes a day for a 50 years smoker, I can bet that the two lungs are not going to look similar. One is going to be all shriveled up and gooey and black. And literally, if you squeeze that lung tissue, you'll be able to squeeze out this tarry substance out of it because of these, long years of chronic tobacco and cigarette smoke exposure. So now you're looking at pathologic anatomy. This is not a normal lung. This is a diseased lung, or a lung from someone who has lung cancer or emphysema or chronic bronchitis. COPD. That's pathologic anatomy, okay? Radiographic anatomy. So of course, a radiograph is an X-ray or a CT scan. So anytime you use radiation to look inside of the human body through the soft tissues into the hard tissues, or sometimes soft tissues are visualized as well, that's radiographic anatomy. So if you're looking at a radiology career or like a CT tech or something, that's what you are. You're looking at radiographic anatomy, okay? All right, so then subdisciplines and branches of physiology. What are those? Cardiovascular physiology, study of the heart, the functioning of the heart, blood vessels, and blood. Neurophysiology, functioning of this nervous system, the central nervous system, which is the brain and the spinal cord, or the peripheral nervous system, which are your nerves. Respiratory physiology, functioning of your lungs, right? And your larynx, your voice box, your trachea, the windpipe, so on. Reproductive physiology, the study of the reproductive structures, testes in males, right? Epididymis and the ejaculatory duct, prostate gland, all of that. In females, ovaries, right? And the fallopian tubes and the uterus and the womb and the birth canal and all of that. Pathophysiology is, again, the study of diseased functioning of the human body. So someone who eats a meal and is non-diabetic, how his or her insulin is controlling the blood sugar levels, and someone who is diabetic, and after having a meal, how is their insulin levels being regulated? That would be an example of pathophysiology. You're studying the physiology of someone who has a disease process going on. All right, let's try to answer these questions. How might knowledge of surface anatomy be important for a healthcare worker during a CPR emergency? So many of you might be CPR certified, right? Or BLS, basic life support, or ACLS, advanced cardiac life support certified. So in order to do a cardiac massage or a CPR on a person, you better know where to do it. So you look for the sternum, for the breastbone here, and then this is how basically you pump up the heart, right? So surface anatomy, you should know the heart is located in a substernal, just under the sternum in that region. What is the relationship between anatomy and physiology? Anatomy is the study of structure, physiology is the study of function, and you cannot study either of them in isolation. They go together. We used the human foot as an example. Dash physiology examines how the heart, blood vessels, and blood function. Cardiovascular physiology would be the answer, right? Okay, so we talked about how anatomy and physiology are basically related. Compare and contrast how anatomists and physiologists specifically describe the small intestine. And an anatomist would be like, all right, so the small intestine is about 6 meters long. It's about 7 inches wide. On the inside of it, on histological examination, you see these finger-like projections called the villi, whose function, not the function, sorry, that's physiology. They also have these indentations. The mucosa is kind of like the columnar type of mucosa here, columnar cells, right? That's what the anatomist would look for. The physiologist would say, all right, so the villi, the finger-like projections are there because they help increase the surface area available for absorption of nutrients from the small intestine. The small intestine basically has these muscular contractions called peristalsis, which push the food from the mouth towards the anus, right? Also, the small intestine secretes some digestive juices to break down the food. This is the domain of the physiologist, okay? All right, so some good practices for how to study effectively for a course, all right? So multiple short study sessions rather than cramming everything in the last few days, never a good thing to do. So break it down into smaller chunks, schedule regular daily study sessions well before the exam. Very good strategy there, minimize distractions. And I basically conditioned myself to work and perform at my optimal level, even with the distractions, because I didn't know what types of circumstances I will be taking my USMLE exam in. So I would deliberately have my sisters make a lot of noise and, somebody else turning on the vacuum cleaner and whatnot, and yet just be able to maintain my focus. So if you find that you're that type of a person, like an intensive training, so be it, right? All the best to you. So and then quizzing yourself repeatedly, working with a partner, flow charts, all those kinds of things. These are great strategies. Why would studying with a partner be more effective? Because then your partner will be able to question you, you will be able to question them, and then they will be able to see things that you missed, and you will be able to see things that they missed. So it's always two heads are better than one, as they say, right? Okay, so let's start with Characteristics of living things. How do you even know what life is, what a living thing is? And the answer is a little more complex than a lot of people think, okay? Uh, because you might say, okay, living things move, all right? But there's many living things that don't move, like sponges, like coral, right? They're attached, they're sessile, which means attached. So, but yet they're living, just as living as me and you. The grass outside or the plants outside, they don't seem to move, but they're living. Okay, so they all eat, they breathe, they excrete, right? But you don't see plants breathing in and out. All right, it's not visible, but they are respiring. So so what are some of the properties that are common to all living things? Organization, number one. What does that mean? Living things are organized from the smallest cells into then tissues, into then organs, and then organ systems, and then organisms. There's a certain hierarchy and organization, which you will not find in non-living things. For example, if you break down and analyze the table or the chair that you're sitting on or the bed, it does not have cells and then into tissues and then organs. It's not a dynamic system. Okay, it's basically wood that has been carved and patterned into some patterns. That's it. So living things are organized. Number one. Uh, all living things metabolize. They have metabolism. What is metabolism? It's chemical reactions taking place inside of that system. All right. So as we speak right now, your cells are taking up food in terms of glucose and burning that glucose with oxygen that you're breathing in through your lungs. Uh, it's happening in every cell of your body, and you're releasing energy in the form of ATP, which keeps you alive. And you're also generating waste products like carbon dioxide and water, which you breathe out or you perspire out or you urinate out or whatever. So that's all living things do it. Now, metabolism is of two types, what is called anabolism and what is called catabolism, okay? What's the difference? Anabolic activities, anabolism is about putting smaller stuff together to make larger stuff. For example, if you eat a lot of protein and you work out, you lift weights, you are incorporating that protein into your muscles to get more muscle mass. That's an anabolic state, right? Or if you eat a lot of fatty foods and you don't exercise and you put on a lot of weight as fat, that is fat anabolism. You're putting on fat, right? But you could be in a negative uh, state as far as your muscle is concerned, because you're putting on fat but not muscle. Your muscles are actually breaking down, right? So then what is catabolic? Breaking down. And this is what happens when you are not eating enough calories and you're expending more calories than you're eating. So then your muscles start to break down, your fat deposits start to go down. That's catabolic breakdown. Okay. Or if you develop a chronic illness like some chronic infection or cancer or something like that, and your body starts to break down, your muscles start to break down. That's called catabolic activity, right? So what you look like right now, your metabolism is an interplay and a balance between anabolic and catabolic activities. If you're more anabolic, you're putting on weight and mass. If you're in a catabolic state, then you're breaking down stuff, okay? And most of our metabolism is basically inherited, all right? It's genetic. So you inherit your metabolic rate from your paternal and your maternal lineages, okay? And so so some people are genetically fast metabolizers. They just seem to sniff food. Uh, I'm sorry, when you say fast they seem to enjoy all kinds of food as much as they want and never put on an ounce of fat. They're still as fit as ever, right? And others are seem to be slow metabolizers. They might just sniff food and turn on packing on the pounds, right? And so a lot of that is also genetic, all right? So it's inherited from your ancestors here. So then you have growth and development. All living things grow and develop. So what's the difference between growth and development? Growth is an increase in size. So you are obviously taller and bigger than what you were when you were 5 years old. Growth. What is development? You're able to solve complex problems now much more efficiently than what you were able to do when you were in grade school, right? Brain development. That's what happened. Or you're able to reproduce now. You can become parents. You could not have parented or given birth to anyone, parented a child when you were 5 years old, okay? Unless it was a case of serious precocious puberty or something. So that's growth and development. All living things respond, okay? Living things respond to external conditions. living things also respond to internal conditions. Okay, for example, if you feel hot, which is an external stimulus, then you might like peel off the clothes or turn on the AC or move to a cooler area, right? So you're responding to an external condition. But if your blood glucose levels drop drastically low because you have not eaten in a while, then you will go and seek out food. So now you're responding to an internal stimulus, hunger, right? Uh, so so these are some examples of, or thirst for that matter. If your body water concentration, your fluid concentration falls below a certain level, your body's own thermostat and food and water regulator, which is the pituitary gland, small pea-sized and pea-shaped gland located at the base of your brain, that will compel you to seek food and water. Okay. All living things regulate. This is extremely important. In other words, all living things are able to maintain homeostasis. What does that word mean? Self-regulation, which means homeostasis. Homeo means same, and stasis means standing still. So same standing still, which means maintaining constant body conditions. In other words, look at your body temperature. What is it? The average body temperature is, core body temperature is 98.7 degrees Fahrenheit, right? Doesn't budge much from that level, whether you're standing out in a freezing Arctic tundra or whether you're out in a blistering desert somewhere, right? Your body will do whatever it needs. You will start shivering and you'll wrap yourselves in warmer clothes when you are in cold conditions, and you start sweating out to cool down when you're in hot conditions. That is homeostasis, maintaining your temperature at that constant 98.7 degrees Fahrenheit. Same with your blood glucose levels or blood water levels. When you're thirsty or famished, you seek food. When you're satiated, uh, uh, you stop eating or drinking, right? If you're satiated at that point. So that's homeostasis, maintaining constant internal conditions. That's a hallmark of all living things. They maintain homeostasis. Reproduction, again, all living things reproduce. They make copies of themselves, either sexually, where you need two partners, two parents, or asexually, okay? So here you're looking at the levels of organization from the simplest uh, to the most complex in living systems, all right? So looking at the hierarchy of organization, as we were mentioning before, for all living systems, this is what it is. It goes all the way from chemical to cellular to tissue to organ system, and then finally to the organismal level, as we see here. Okay, so what does that mean? When you talk about the chemical level, what is the very chemical basis of life? All living things are made up of what we see here, atoms and molecules, as are all non-living things as well. So uh, all living things have like sodium, potassium, calcium, magnesium, so on. Those ions there, water molecules, of course, carbon dioxide, right? Uh, as do many non-living things. So chemical level is something which is not specific or unique to living things. However, when you get to the next level here, which is cellular level, that means what? Cells. Now, cells, once you get to that level, you're talking about a living thing for sure, right? Non-living things do not have a cellular composition. So what is a cell? As you can see here, it's the basic unit of life, okay? Also called the smallest unit of life or the structural and functional unit of life. That's the definition that I tend to prefer, all right? The cell is the structural and functional unit of life. In other words, I can simply extract a skin cell from your forearm or your forehead or someplace, put it in a petri dish, give it the right growth factors and nutrients and oxygen, and come back and check on it in about a couple of weeks, and I might have a whole new beautiful sheet of new skin growing, right? In other words, the cell is able to replicate, to reproduce like all living things. They need nutrition, they need respiration, all of that. So these are the smallest, uh, simplest, and structural and functional units of life, all right? Then cells come together, similar shaped and similarly functioning cells to form tissues. Tissues come together to form organs, right? Organs come together to form organ systems. And then, of course, organ systems come together to form an organism. All right, so this is basically what we are looking at here. So here in this picture, what you're seeing is a single atom, okay? Looks like that is a hydrogen atom, the simplest atom in existence. And how did it occur to me that this is the hydrogen atom? If you look at the next example, a molecule, and a molecule is two or more atoms joined chemically together. So it looks like an H2O molecule, right? Uh, so two hydrogens, and just saw a rabbit dart by in my backyard. So it was pretty cool. This nice weather brings out the rabbits here. So let's make a quick note. Saw a rabbit dart in backyard. So that tells you the importance of keeping up with the lectures and listening to them, and the notes, of course. So you never know what's going to pop up on your extra credit exam, extra credit questions on the exam. That is okay. So a single hydrogen, I'm sorry, a single oxygen and then two hydrogens. So H2O, that's a molecule, water. And no life is possible without water. That's the fundamental, the most important inorganic substance of life, okay? And we'll talk about what is organic versus what is inorganic. Molecules come together to form macromolecules or large molecules, such as the one we see here. That's, everyone knows what this is. This is a double helix DNA strand. These macromolecules come together to form organelles or mini organs, as they're known as, like the nucleus of the cell, endoplasmic reticulum, Golgi apparatus, peroxisome, so on and so forth. And then the organelles come together to form a cell. Here's a cell. This is the first structure where life is in existence now, all right? These things are the precursors to life, but not living in of themselves. But here, a cell, that's a living entity. So then the cells come together to form a tissue, as you can see here, and I can tell you that this is a columnar epithelium. It's a ciliated columnar epithelium. And so I can tell you that it's taken from your respiratory tract, okay, just based on the picture. So the cilia and the columnar epithelium and the underlying connective tissue here, this is what your upper respiratory tracts, like your trachea, the windpipe, and the bronchi look like, okay? That's where this is likely taken from. So in any case, the tissues come together to form an organ, like your intestines. That's the large intestine, which is kind of covering like a drape, your small intestine in the middle, right here. And then these organs come together to form organ systems, like what organ system are we looking at here? The digestive system. You can see the esophagus, the stomach, large intestine, small intestine, so on and so forth. So that's the digestive system, and then a full organism. Okay, so there's a definite hierarchy that you see. Okay, now you're looking at the different organ systems in the human body, 11 of those. Now, the first 10 are basically the same in all humans, regardless of their biological sex. To look at the 11th one here, male and female reproductive systems, now they are anatomically and physiologically different depending on whether you are a biological male or a genotypic female, right? Or rather phenotypic. We'll talk about genotype, phenotype later when we get into genetics as well. All right, so let's take a look at these systems, what these are. So first you're looking at the integumentary system. That integument means a covering. So what is our body covering? Also the largest organ in the human body, your skin, of course. So the skin and the associated structures like sweat glands, also called sudoriferous glands, sebaceous glands, which secrete oil, hair follicles, nails, and hair. All of these are parts of your integumentary system, as you can see here in this picture. All right, then the skeletal system, of course, made up of 206 bones, and we'll get into that as well in detail. The muscular system, here are all of the muscles, 650 or so muscles of the human body. Nervous system, which is made up of, comprised of a central nervous system, which includes your brain and the spinal cord, and the peripheral nervous system, which includes the peripheral nerves, which supply sensation and action to the rest of the body. All right, endocrine system here is made up of glands and hormones. All right, so all of the glands, glands are those organs that secrete chemical messengers called hormones here. Then the cardiovascular system is made up of the heart. In red, you see the arteries, and in blue, you see the veins, and then capillaries as well. Then you have the lymphatic system. So these are lymphatic ducts, lymphatic trunks, lymphatic vessels that carry this milky white fluid called lymph, which is derived from blood, okay? Then you have the respiratory system. So the lungs, the windpipe, bronchi, trachea, the larynx, all of that. Here is the urinary system made of the two kidneys, the ureters, the urinary bladder, the urethra. Here is the digestive system, again, from the mouth, the oral cavity to the pharynx, down through the gullet or the esophagus, stomach, small intestine, large intestine, rectum, anus, and out. All right, then you have the male versus the female reproductive system. Male reproductive system has testes, and females have ovaries as gonads or sex glands. All right, so what does it mean if an organism is responsive? Well, if an organism is reacting to either external or internal stimuli, it's responsive, right? Patients too. For example, if you work in the ICU, the medical ICU or the surgical ICU, by the way, that happened to be my very first medical rotation. So when I started my medical residency, my very first medical rotation was in the medical ICU, which was my very first residency rotation. And so as my dad told me, and my dad also is a practicing physician still, okay? So let's say my dad's analogy. Yeah. All right, so what he told me was, and I had a choice as to what electives, what rotations I could choose. So I went for medical ICU straight up, first one, the most intensive medical rotation of all, arguably. And so my dad said, well, son, now you are in the eye of the storm. So that's how you're starting out, all right? And indeed, that's what it turned out to be, quite intensive, but at the same time, I learned most in that intense environment during my medical ICU rotation about critical management of patients than in any other rotation. So it was all good. How does this characteristic relate to the survival of this organism? It can escape from danger, and it can gain nutrition and shelter, and it can reproduce if it's responsive. Does a higher level of organization contain all the levels beneath it? Yes. Which body system, organ system is responsible for filtering the blood and removing the waste products of the blood in the form of urine? The urinary system, of course. All right. So now what we are looking at is something called the anatomic position. And this is extremely important and pertinent in the medical field, because whenever you describe a patient's pathology or position or location of an injury, a wound, a cut, any kind of an anomaly, right? You always automatically assume that you're referring to the patient in the anatomical position. It's for reference terms, right? So for example, if you say that a person has a cut in the right hypochondriac region, right? What would that be? You automatically have to conjure an image of a person standing like this lady in the picture, all right? So what is the anatomical position? You stand straight, looking straight forwards with your palms facing forwards. Look at the palms of the lady in question here, forwards, right? Your feet slightly apart, about the level of your shoulders, shoulder apart, right? And you're looking straight up. That is the anatomical position, and that's the default setting for all references to the human body. Just remember that, okay? You're not gonna visualize someone hanging upside down or standing lopsided or showing their back or something like that. That's not the anatomical position. By definition, you're looking at someone who is standing upright, facing forwards, palms forwards, toes facing forwards, as in this picture, all right? So that's the anatomical position. And that was one thing that was emphasized over and over again during our anatomy lessons in first year of med school. Okay, our models, sometimes they'll come and deliberately they're like pose, someone showing the muscles or six pack or standing like some hero out of an old western or something. So the first thing that we were taught to do was correct the posture of the model slash patient. They should be in the anatomical position, all right? So for reference, again, you're not going to turn your actual patients, force them into the anatomical position. So, all right, so why is the anatomical position pertinent in other ways? Well, because that gives you access to what are called planes and sections, all right? So, all right, so what are the sections or the planes for that matter? So let's start with the first one, which is called the coronal plane, or the coronal section. So the coronal, also called the frontal plane or the frontal section, okay? So what you do here is, as you can see in this picture, you visualize this lady or any human being for that matter being transected or cut in this way, right? So if I put my palm like I am here right in the middle of my skull, top of my head, and cut all the way across, what's going to happen? My front is going to fall in the front, and my back portion is going to fall in the back, right? So in other words, what a frontal section did was divide the body into an anterior, which means the front, and a posterior, which means the back portion. Okay, that's what a coronal slash frontal section is, and you can see it here. That's a coronal plane. If you cut the lady in this way, you're going to come across the coronal section. And here's the coronal section of the skull, and you're likely looking at a CT scan, right? Computerized tomographic scan or computerized axial tomography is what you're looking at here. You're looking at the brain, the eye sockets, the nasal area, the maxilla, and all of that, right? This is the coronal section. So even if you're working as a radiologist in the radiology department, you better know your planes and sections because that's how you're going to look at pictures. All right, so then let's take a look at the mid-sagittal or the sagittal section, okay? So what we have here, look at the mid-sagittal section, it's this one. Now, if I were to cut myself right in the middle from front to back, this way, right? Right, right across like this. So instead of cutting this way, I'm going right here. So what's gonna happen if I cut myself this way, straight in the middle, my right side is going to fall to the right and my left side of the body is going to fall to the left, that way, right? This is your sagittal plane or sagittal section. So what a sagittal section gives you is a right and a left profile, the two sides, okay? Right versus left, that's sagittal section. Even the word sagitta means arrow. So for example, your zodiac sign, if you're a Sagittarius, we all know what the zodiac sign, or I think we all know, maybe you're not into zodiac signs. So zodiac sign, if you talk about Sagittarius, what it says is, what it shows as a symbol is a half human and half horse, right? What were they called? Not minotaurs, centaurs, yes. Centaurs were like half humans and half horses in Greek mythology, right? Centaurs. So Sagittarius, because the human here in question is actually carrying a bow and arrow, right? So Sagitta means an arrow. So if you shoot someone with an arrow right in the middle, you split him into half, right and left equal halves. That's the sagittal section. If it's exactly in the middle, it's called a mid-sagittal section, as it states here, right? But if you do not cut them in the middle, but it's rather a little bit tilted to the right or left, then it's called a parasagittal. Para means surrounding. So a little more on the left or tilted on one side, that's a parasagittal section. Okay, transverse section, finally, what is that? If I were to take a sharp object and cut myself this way. So my top is going to detach and my bottom is going to detach as well, right? So in other words, here you're looking at the brain, when a transverse section was carried out, and so the top of the skull was moved, so you have a superior versus an inferior view of the skull cavity. There you go. Coronal section, anterior and posterior, back and front, sagittal section or mid-sagittal, right and left halves, and transverse section, superior and inferior. Okay, very important to remember for radiological studies, of course. All right. So here the same thing can be seen with the intestines, as you can see. Now, there's another one called the oblique plane. An oblique plane is, as the name suggests, it's slightly inclined. So if you cut some structure at a tilt, at an angle, like if I were to go across the lady this way, that would be an oblique plane, okay? An oblique cut, right? So, okay, now here in these pictures, you can actually appreciate these anatomical terms, what those mean, right? So look at the guy here. So the model in question, the front is called the anterior, the back is called the posterior. So the navel or the belly button, right? The umbilicus is anterior, while the gluteus maximus, the hip muscle or the butt muscle, is posterior. Another term for anterior is ventral. Another term for posterior is dorsal, like the dorsal fin of a shark, one of the most feared sights in the natural world, right? Okay, superior and inferior. Superior is higher up, like the head is superior to the foot, obviously, as you can see. So superior versus inferior. All right, medial and lateral. Medial, if the structure is closer to the midline, like the umbilicus, the belly button is medial to the arms, right? Or the nose is medial to the ears. Or conversely, the ears are lateral or sideways to the nose. The nose is medial to the ears, right? And the ears are lateral to the nose. So medial towards the midline, lateral sideways. Okay, then proximal and distal. So these terms actually apply to your limbs, your upper limbs and your lower limbs. Okay, so what does it mean? Proximal means it's closer or in close proximity to the attachment. Distal means it's more distant, away from the point of attachment. For example, the shoulder joint is proximal to the elbow joint, correct? Because the shoulder joint is attached with the point of attachment. So the shoulder joint is proximal to the elbow joint. The elbow joint is proximal, closer to the attachment as compared to the wrist joint, right? And then the wrist joint is distal to the elbow joint, farther away from the attachment than the elbow. And the wrist is certainly distal to the shoulder joint as well. So proximal versus distal. Proximal closer to the attachment, distal uh, farther away. Okay, so axial versus appendicular region. What is the meaning behind those? Well, axial, the word comes from axis, and axis means the midline, the center point of anything, right? So this is the axial plane or the axial structure, the midline, which would be the spine of the person, right? The skull and the spine. And then appendicular, that word comes from appendage, which means something which is attached to the middle. So that would be this. These are the appendages, the appendicular plane, the limbs, upper limbs, and the lower limbs. Okay. All right. So all right, next up, we are looking at regional terms. Now here, there's some memorization that you guys will have to do, okay? Some of these terms are self-explanatory, some of them are not. So you basically have to just memorize, go over this stuff. And when you do your labs, this is going to become more clear as well there, okay? All right, so taking a look here, let me just make sure. Okay, so cephalic. Cephalic means the head, cephaly, head, right here, okay? Frontal, the forehead, frontal region. Orbital, the orbits of the eyes, so the eyeballs right here, the sockets of the eyeballs, right? Buccal, cheek, cheek. Oral, mouth. Mental, in this case, is your chin. Not mental as in brain, but chin, okay? Thoracic is, of course, the thorax, the chest region called thoracic. Axillary region is your armpit region, right? Axillary. Memory has to do with your breast, the chest region right here. Pectoral has to do again with the chest, the interior, the front of the chest. The sternal is with your breastbone or sternum right here, okay? Then the pelvic region is in the pelvis right here. That's where you have your reproductive organs and your urinary bladder, right? The coxa or the coxal region is the hip region. This is coxal. Inguinal region is the groin region, which is here. So memorize those terms. Cervical, from cervix, neck. Cervix is Latin or slash Greek for uh, the neck. So there's another cervix other than this neck. The cervix in a female, right? The cervix of the uterus, uh, where you do a pap smear, as an example. So, but the term cervix actually means the neck. All right, then the upper extremity. Here's your shoulder, called the deltoid region. So this muscle is also called the deltoid muscle, right? Then the brachial is your arm. Brachial is for the upper arm. Antebrachial is your elbow, the front of the elbow. Then the antebrachial is the forearm. Carpal, carpus is your wrist. Manus, like manual, manus, hand. Palmar, the palms, of course. Digital, digits, fingers. Similarly, in the lower extremities, the femoral region is the thigh. Thigh is femoral, right? Like the femoral bone. Femur is the bone, the thigh bone, or the femoral artery, one of the major arteries of the human body. Patella, or the patellar region, is the kneecap here. Popliteal is the back side of the knee, the posterior or the dorsal side of the knee. Crural, lower leg. Sural, for the calf in the back. Calcaneal, heel of the foot. Plantar, the sole of the foot, because the plantar surface is what you plant on the ground, the soles of the feet, okay? Then the ankle region is called the tarsal region. Carpal for the wrist, tarsal for the ankle. Then dorsum of the foot, the top of the foot, and digital, the toes there. Pes for foot. Pes is the term for foot, okay? So same, now you're looking at the posterior end of the person and see, notice how he is in the anatomical position, right? Something to observe here. So same structures that you can see here, same terms. Here in the back, you can also see the vertebral region next to the vertebral column, the backbone, the lumbar region, okay, right here, lower back, sacral region, the small of the back, the gluteal region from that's the buttocks, like for gluteus maximus muscle, the perineal region right over here, okay, and the other regions that we talked about before. All right, so here what we are looking at is something called your body cavities, okay, and you can see them both anteriorly in the front, and you can also see them posteriorly in the back. So let's take a look at what is the anterior situation as far as these cavities are concerned. So first up here, you have the thoracic cavity, the thorax, the chest. Now the thorax is separated from your abdominal cavity down here by a dome-shaped skeletal muscle called the diaphragm. Here's the diaphragm or the thoracic diaphragm. This muscle demarcates your chest or your thorax from your abdomen anatomically. This is the demarcation, okay? And what you find here, of course, in your thoracic cavity are the two lungs, as you can see here. And then you have the heart in the middle. You can also see, what is that? The sternum, the breastbone is also peeking out. And then, of course, here's your diaphragm, the muscle that separates the chest from the abdomen. And in the abdomen, what you have is digestive organs. Of course, you have the stomach, spleen, pancreas, small intestine, large intestine, right? Even blood vessels like the abdominal aorta, the inferior vena cava, all of those structures are found here, okay? So these are your anterior body cavities, okay? Now all these cavities are filled up with some fluid, a serous fluid. Serous means watery, thin consistency like water, watery fluid. And this fluid is very important because it prevents friction in between the organs and it allows for the organs to expand and constrict, like your stomach. When you eat, it expands and then it constricts. Same with your heart, it's pumping, beating, so it's expanding, contracting. So to prevent friction with the surrounding tissue, you have these fluids here. All right, how about the posterior cavities? Here you have something called the cranial cavity. And this, of course, encloses your brain. And it's filled up with cerebrospinal fluid around the brain. And then it continues downwards as the vertebral cavity. All right, so these two are posterior in the back, and they're filled up with the cerebrospinal fluid again, okay? All right, so what we are looking at here is that the fact that all of your organs are actually surrounded by membranes, protective membranes, okay? And they are, the best analogy that I can give you here for your organs is if you were to insert your fist, as you see here in this picture, into a balloon, all right? So your fist itself is an organ. Think of it as a stomach or your lungs or your heart or whatever, right? The fist itself. This is the membrane surrounding your heart, and this outer layer would be called the parietal layer of the uh, pericardium. Pericardium, peri means surrounding, cardium, heart, as an example. So pericardium uh, has two layers. One is a parietal layer, which is this layer, but you also see that there's an inner layer, this one, right? Right next to your palm, your fist here. And so this inner layer would be called the visceral layer of uh, the visceral layer of the pericardium. If this is a heart, this could be a lung too. If this is, if you're talking about a lung, if this would, you consider this to be a lung, then this would be the parietal layer of the pleura. Pleura is the membrane that surrounds the lungs, and this would be the visceral layer surrounding of the pleura. Okay, so something to remember. The serous membranes surround all your major vital organs, and the serous membranes are filled up with a fluid called the serous fluid. That's what we're looking at here, okay? So right? Posterior and ventral cavity, we talked about those, right? Uh, what they have here, parietal and the visceral layers of the serous membrane is what we are seeing. And they're there for prevention of friction, allowing for the organs to distend and contract again. All right. So mediastinum, medi means middle. All right, estinum chest, so middle of the chest. Mediastinum is literally what we're looking at. So this region is packed with important anatomical organs like the heart, the lungs, thymus, which is an important endocrine gland, your windpipe or trachea, blood vessels, all those kinds of things. Then the heart is surrounded by the pericardium, this serous membrane, as we mentioned before. The outer layer is called the parietal pericardium, and this layer doubles up and folds in onto itself to form the visceral pericardium, which is closely attached to the heart itself. And in between the parietal and the visceral uh, pericardium, like this, in between the parietal layer and the visceral layer is fluid. You see that this would be fluid. And so in this case, this would be called the pericardial fluid, the pericardial cavity in between the parietal and the serous layers of the membrane, and they are filled, it's filled up with pericardial fluid. Same with your lungs, parietal pleura here. So this is your pericardium, right? So that's your parietal pericardium, the outer layer, and see how it folds onto itself to become the visceral pericardium right next to the heart. And you can see that in between these two, the parietal and the visceral pericardium, this bluish pericardial fluid is located at right here, right? Same with your lungs. Look at the lungs. So here is your parietal pleura, the outer membrane. It folds onto itself to form an inner layer called the visceral pleura, right next to the lung tissue itself. And in between the parietal and the visceral pleura is a pleural cavity, which is filled up with a pleural fluid because lungs also expand and contract with each breath that you take. So to prevent friction, all kinds of things, you have that. Okay, so here, right, we are looking at the peritoneum, okay? And the peritoneum is your abdominal cavity, the membrane, the serous membrane surrounding your abdominal cavity, okay? So the peritoneum, needless to say, is an extremely important area because the peritoneum has all of your important digestive organs, as you can see here, right? And then again, what we have here is an outer parietal peritoneal layer, the parietal peritoneum, the outer layer you see here, all right, of a serous membrane. And then it folds onto itself to form here the visceral layer of the peritoneal cavity or the peritoneal membrane. In between the parietal and the visceral peritoneum is this peritoneal cavity, which is filled up with peritoneal fluid, okay? So now this is an extremely important area because when I worked in the surgical unit during my medical rotations, a fair number of cases that I saw there came in with conditions that had to do with the peritoneal cavity. Conditions like um, appendicitis, acute appendicitis, conditions like hernias, inguinal hernias, paraumbilical hernias, conditions like gunshot wounds, conditions like stab wounds, like gastric ulcers, perforated gastric ulcers that had blown open, right? Uh, Peptic ulcers, abdominal cancers, uh, stomach cancer, pancreatic cancer. So for all of those conditions, you have to get cut into the peritoneal cavity. So to learn about those structures, right? So let's see, my tenure in the surgical ward and peritoneal cases like acute appendicitis, even ectopic pregnancy, right? Ectopic pregnancy, cesarean sections as well, uh, perforated peptic ulcers, okay, gunshot wounds, stab wounds, C-sections, ectopic pregnancy, etc. Okay. Okay, so why am I mentioning all these conditions? Because here's the deal. anytime that there is a puncture in your peritoneum, either external, such as in a gunshot wound or a stab wound or any other penetrating injury into your peritoneum, or an internal rupture like a burst appendix, burst stomach, burst pregnancy like an ectopic pregnancy or whatever, anytime that happens, then you end up with something called peritonitis, which is a serious life-threatening infection of the peritoneal cavity. All right. So peritonitis, how would you diagnose a patient with peritonitis? So let's go over the steps. A patient is wheeled in. You are working in the ER, right? Or you're working on the medical floors or the surgical floors, and this patient, they ask you down for a consult to take a look at this patient. The patient was in a barroom brawl, a fight in a barroom as an example, and he fell on like a sharp, on the sharp corner of a table and got this penetrating injury, or maybe he was stabbed in the stomach or something like that in the abdomen, right? And so the person comes in bleeding profusely from the wound in the abdominal region, let's say in the umbilical region, right? The patient is tachypnic, like breathing fast and shallow, right? Is diaphoretic, which means is sweating. And let me quickly write this down too. Let's say example of barroom brawl victim in the ER and peritonitis, signs and symptoms, and diagnosis and treatment. Okay, so right, so the person is profusely sweating, is diaphoretic, is like kind of confused and disoriented, right? Uh, so you do your vital signs. What are the vital signs? There's 4 vital signs, right? Every good nurse, every good medical student should know this. So the 4 vital signs are the respiratory rate, right? The rate at which the person is breathing, their temperature, their pulse, and their blood pressure. So let's take a look at our stab wound, barroom brawl victim here, okay? So the pulse is rapid and thready, fast pulse, but thready, okay? The skin is cool to the touch. The blood pressure is 90 over 60 millimeters of mercury and dropping, okay? The temperature is 97 degrees Fahrenheit, or maybe slightly elevated, let's say about 99.2 degrees Fahrenheit, okay? The respiratory rate, as I mentioned, is increased, okay? Breathing at, let's say, 25 breaths per minute. All of these vital signs are pointing in the direction of what? Rapid volume loss or blood loss. This patient is crashing, is going into circulatory shock, okay? So in any case, when you do a medical exam, and which you do after a medical history, so you have to take a full history of this patient. If the patient is able to answer questions themselves, ask him, what happened? What time? How did you feel? Uh, where was the pain experienced? Are you still experiencing pain? How bad is this pain on a scale of 1 to 10, 1 being the least and the 10 being the worst type of pain that you've ever experienced, right? Is the pain kind of a dull, achy type of pain, or is it a sharp electrical pain, or is it like a constant boring pain, right? Does the pain stay in one place, or is it radiating? It's moving elsewhere. Are there any factors that make the pain better, that alleviate it? Are there any factors that make the pain worse? So you're going to ask all these questions, right? Any travel history, any surgical history, any medications that you are taking, right? All of that. If the patient is non-vocal for whatever reason, ask the attendants, the people who brought them in, people who were witness to the incident, right? Get as much information as you can. A good history is critical. Everything builds on that. If you cannot take a good history, you're at a tremendous disadvantage. So then you move on to the physical examination, okay? So when you go to the medical examination, what do? There's 4 things: inspection, palpation, percussion, and auscultation. Okay. So what are those? Inspection. Inspect, visually look at the patient. You might have to disrobe, unclothe the patient. They're lying on a gurney or on a stretcher or a table or whatever, right? Or sometimes they would be standing upright. Uh, And then so you will be taking a looking for things like obvious bruises, cuts, scrapes, wounds, swellings, tumors, rash, yellowish discoloration of the skin, such as in jaundice, or redness of the skin, something called erythema, right? Um, any skin discoloration, moles, those kinds of things, skin markings. So that's your inspection, very important. So in this patient, obviously, you see when you look at the abdomen, there's this gaping wound in the paraumbilical region, okay, which is basically oozing out blood. All right, so then you move on to palpation. What is palpation? You will gently touch the body surface of the patient, checking for any underlying uh, swelling or tumor. Tumor is a swelling, basically, or any pain. If you touch an area and the patient winces or just doesn't let you go there, there's obvious pain there. All right. So palpation, then percussion. And percussion is what? If you've ever played the drums or the bongos or any of those instruments, that's what percussion is. Percussion is when you tap, take your left palm flat, put it on the body surface. Okay, I'm going to do it on myself here, and I highly encourage each of you to do it on yourselves or your partners or whoever volunteers to be your clinical case. Okay, so here, uh, put the left palm flat here, and I'm palpating the lungs, the chest cavity, the thoracic cavity, obviously, right? And using your right hand, this is going to be my hammer, okay? My middle finger of my right hand. I'm gonna tap the uh, the distal interphalangeal joint, which means this knuckle, the top knuckle over here, all right? And I'm gonna tap the middle finger again. Middle finger is the hammer, and the left middle finger is the area where I'm gonna tap. So I don't know how clearly you heard that, but it was a pretty like a hollow note, like a resonant note. Okay. Now, so this is now let me palpate here on my breastbone or my sternum. Yeah, so I'm going to percuss rather, not palpate, percuss here, right? All right. So now I'm getting a very dull, stony, solid note. Okay. What did it just tell me? So, for example, if I percuss here on the laptop, mm, semi-solid, right? If I percuss on the wall here, all right, kind of hollow, okay? So what this percussion is telling me is whether the underlying area or the underlying organ is hollow, filled up with air, or if there's a solid mass, a tumor, and foreign object or something down there, okay? So that is percussion. So if the patient is not in that much distress or pain, and if they allow you to do the percussion, percuss. So for example, if you're percussing the lungs, right? And so what do you expect to hear? What type of note if you're percussing the lungs? Well, the lungs are air-filled hollow organs. They're hollow, right? Air-filled. It should be a hollow note. But what if you are percussing your lungs, especially the base of the lungs down here, and you're picking up a solid note? What does that tell you? There's something underlying, either a solid mass in the lungs or underlying fluid. There's fluid in the lungs. What can cause fluid in the lungs? Pneumonia can, right? Tuberculosis can. Uh, Sometimes uh, pleural effusion, water surrounding your lungs, following a bad lung infection, right? Or mucus, too much mucus there. So what can cause a solid object kind of sensation there? A tumor, a lung cancer can cause it, right? Or any other mass that is growing there. So percussion will tell you a whole lot uh, in terms of what lies beneath. Okay, so one of my all-time favorite movies as well. It was titled "What Lies Beneath." So let's take some notes here. Okay, so percussion and all right. So so what did we do so far? Inspection. We did palpation. The patient has obvious tenderness and pain in the region where there's the stab wound, right? Then we did percussion. So when you uh, percuss here. So if the patient has developed peritonitis, which means the stab wound or whatever organ ruptured has caused infected material to spill out from the guts or from the internal organs out into the peritoneal cavity. Now the patient has peritonitis, a full-blown infection of the abdominal cavity. That is life-threatening. No time to waste. This person needs to be operated upon. A laparotomy, which is like a full body opening up procedure, cutting into the body. Laparotomy is what is done, what is needed at this point. Okay, so when you palpate someone with peritonitis, extreme pain, extreme pain. All right. So this patient with peritonitis will demonstrate something called abdominal rigidity or guarding, okay, which means what? As soon as you touch the abdominal region near the stab wound, the patient winces in pain and tenses the muscles of the abdomen. You can feel it, he's tensing him, right? So this is also called guarding. So guarding and rigidity tells you there's pain, significant amounts of pain there, right? So that's what you're going to get on palpation. Then on percussion, if the patient even allows you to do that, because there's so much pain, if you percuss and you're picking up, what are you picking up here? Solid notes from the intestines, and you know, intestines should be hollow, right? They're air-filled organs. Sometimes there's like food and water in there as well. But if you're getting a solid note throughout the intestines, there's something called intestinal obstruction going on. The intestines are not moving, right? They're not moving. They're filled up with either blood or other fecal matter or whatever. And then finally, you go to the fourth step of physical exam, which is auscultation. All right. So and I'm going to write these down quickly here as well. Inspection. We said what? We said palpation. Then we said percussion, and now auscultation. So what is auscultation? Auscultation is taking the instrument called a stethoscope and taking a listen to the patient's body, right? That's what auscultation is all about. So in this patient, when you take a listen to the abdomen, okay, what you will come across, or rather hear, is something called a silent abdomen. Okay, so what is that all about? Silent abdomen. Normally, if your intestines are functioning as they should, no problems, and you take a stethoscope and you listen, where do you put the stethoscope, the diaphragm of the stethoscope anyway, to take a listen? Ideally, it's done here in your left lower quadrant, all right? So about 2 inches below your belly button on the left-hand side, okay? Every 30 to 60 seconds, you should hear like this tinkling sound through your stethoscope. And this tinkling is a sign of water and fluids and gas and food moving through your intestines. So these tinkling sounds, abdominal sounds, are normal. In someone with peritonitis, because of some rupture, external or internal rupture, and infection of the abdomen, you will hear nothing, a silent abdomen, right? No tinkling sound, nothing. And this silence is ominous. This is telling you that the person has developed a paralyzed intestine, something called paralytic ileus, all right? And so peritonitis. So now that you have all of these ominous physical exam signs, you could take a few quick diagnostic tests there, such as a chest x-ray, an abdominal x-ray as well, possibly a CT to see the extent of the injury, but you have to take this patient to the surgical suite as soon as possible, because what they are going to do is they're going to perform a laparotomy on this patient. Laparotomy, which means open surgery, right? They're going to open up the abdomen. They will have to stop the blood flow first, all right? And they have to start transfusing the patient at the same time. So arrange, cross-match, cross-type the blood, and start pumping blood into this patient. Blood transfusion, you need it to maintain the vital signs, the blood pressure, the vital stats of this patient. At the same time, the doctor will clean the area, the bloodied area, using suction, okay? So suck out the blood so you have a clear field of vision, and then you are going to patch up the wound, okay? Sometimes it needs surgical adhesives, like literally tape, scotch tape, to seal that area. Or if it's an artery, then you have to clip the artery to prevent bleeding. Now at the same time, all right, so this is called hemostasis, stopping bleeding, hemostasis, right? Hemostasis, blood transfusions. That's what you've done so far, right? And what else? Something called an antibiotic lavage. Antibiotic lavage. So what is an antibiotic lavage? It's just like taking a garden hose and how you water your plants. You're going to take a surgical hose and you're going to fill a container with a broad spectrum antibiotic, all right, which covers most strains of infectious bacteria, which are common. And you're going to literally wash out the intestines, the insides of this person's abdominal cavity, just wash them out. To prevent infection. Okay, that's called an antibiotic lavage, and it's one of the most commonly used antibiotics, broad spectrum antibiotics in the hospital setting, which I used a whole lot, is something called Zosyn. Zosyn, which is the brand name for uh, for a type of penicillin. Zosyn is a type of penicillin, so that would be piperacillin plus clavulanic acid. That's what Zosyn is. Piperacillin plus, it's a type of penicillin plus clavulanic acid. All right. Okay. That's how you would try to save the life of someone who has developed peritonitis, and how to diagnose that person as well. Okay. So next up here, you are looking at the different abdominal pelvic regions and quadrants. This is also important because this will help you understand and visualize what you're up against. Okay. So let's take a look at what that's all about here. So what we have done here is we are looking at someone in the anatomical position, obviously, as you can see, and we have drawn a number of imaginary lines across here. All right, so you have two vertical lines here, those two, and two horizontal lines to kind of make this 9 quadrant picture that you see. All right, so these two are what are called subscapular lines. All right, so you draw these imaginary lines from right here. So they should go through the middle of your collarbone, the clavicle right here. So the right and the left clavicle. So mid-clavicular line, right, just in the midpoint of the two clavicles. And they go straight down on both right and left-hand sides, as you can see. They should bisect your nipple, cut across the nipple, go all the way down straight here, right? And then go right across your iliac crest, which is kind of like your love handles region, all right, to help you understand, and all the way down on the two sides, all right. Then as far as the horizontal lines are concerned, the first one is called a subcostal line. Sub means below, costal means your ribs. So subcostal line is just under the ribs, okay? So here it is, a subcostal line, just grazing your ribs here. And then what is called a transiliac line, because it is going across the two iliac crests, the two love handle bones, if you will, here. All right. So this is what it is. Two mid-clavicular lines, vertical ones, is subcostal line here under the ribs, and a transiliac line across the two iliac crests, these bones here. So what do you end up with after you do that? These 9 regions, okay? And they're named as they are. The right hypochondriac region, okay? Hypo means below, again. Chondriac, the cartilage. Chondra is for cartilage. So you're looking at rib cartilage here. This is bone for the ribs. This area in white is the cartilage. So the right hypochondriac region literally means the area below the costal cartilage or the rib cartilage here, both on the left and the right hand side. Okay. Then you have in the middle something called the epigastric region. Epi means before. Gastric is your stomach. So here's your stomach. So just before the stomach, epigastric, right? Then here you have the right and the left lumbar regions. Lumbar are to the sides of the abdomen. In the middle is your belly button, your navel, also called the umbilicus. So therefore, this is the umbilical region here. Then you have a right and a left iliac region down there where your groin is, right, in the iliac region. And then in the middle, you have something called the hypogastric region. Hypo means below, again, gastric, the stomach. So that was before the stomach, epigastric. This one is hypogastric region, okay? Now, what organs are in each? Just take a look. Look at the right hypochondriac region. What do you expect to find here? The big, massive, bulky liver, of course. That's liver, the largest internal organ that you have, about 3 pounds of tissue. So the liver is for the most part filling up your right hypochondriac region right here. Also the gallbladder, which is peeking out here from below the liver, would be here in the right hypochondriac and partly in the epigastric region as well. So what's in the epigastric region? Again, much of the liver and some parts of the stomach as well. How about the left hypochondriac region? Stomach, you see it? And some parts of the liver as well, all right? You'll also have the spleen and the pancreas, roughly in the epigastric to the left hypochondriac regions. Okay, what do you have in the right lumbar region here? Your large intestine, and this is the ascending colon. Ascending colon. Okay, colon is the other name uh, for your large intestine. So the ascending colon is here. Okay, so then what is in your left lumbar region? The descending colon. So the colon ascends or climbs up here, then it goes transversely in this region called the transverse colon, and then it descends here to form the descending colon. And then it forms a sigmoid colon, and then the rectum, and then the anus. Okay. All right, how about the umbilical region? Again, in the umbilical region, you see the transverse colon hanging like a drape or a curtain at the top. And then you see much of your small intestine located here in the umbilical region, right? Okay, how about your right iliac region? Again, portions of your what? Of your ascending colon and your appendix and your cecum, important appendix, right? Surgeons make a lot of money operating and cutting that little organ out here. That would be found here in the right iliac region too. How about the left iliac region? Descending colon, the sigmoid colon. So those parts of the large intestine would be found in the left iliac region. How about the hypogastric region? What do you see here? Your urinary bladder is there. And also the ovaries in females and uh, the testes are roughly there in the hypogastric region, okay? So this gives you a visual idea of what those 9 regions are and what organs are found in each, all right? So if a patient is complaining of pain in the epigastric region, right, the point, oh, doctor, it hurts here, what are you gonna think? Maybe some liver pathology like hepatitis, or some stomach pathology like gastric ulcer or something, right? So there, if someone is talking about pain in their right hypochondriac region, liver conditions like hepatitis, cirrhosis, so on, or gallbladder conditions like cholecystitis, infection of the gallbladder, something like that. If someone is complaining of pain in the hypogastric region, urinary stones, or UTI, urinary tract infection, or something like that, right? So this gives you a good idea of what organs are found there and what pathologies to think of, okay? So then, okay, so now here is a much simpler way of doing the same thing. Instead of conjuring up those 4 different lines, the 2 mid-clavicular and the subcostal and the transiliac line, you can simply make like a bullseye here, right? Only 2 lines. So this is a substernal line. It starts from below the sternum, your breastbone, all the way here. It bisects or goes across your umbilicus, your belly button, and goes all the way down to your pubic symphysis area here, straight line, right? And then you also have a horizontal line, which again is kind of like a transiliac line in between the 2 iliac crests here, all right? So now this is a bullseye situation, and you end up having only 4 quadrants versus the 9 regions in the first one. Here you only have 4 quadrants, a right upper, left upper, right lower, and a left lower quadrant. This is simpler, right? But each quadrant also has more organs now. It's not as specific as the 9 regions that you saw, but it's simpler to visualize, all right? So for example, in the right upper quadrant, if someone says they have pain there, you have to think of the liver, you have to think of gallbladder pathologies. You have to think of the ascending colon or transverse colon pathologies. You can also look at the stomach conditions like gastric ulcers, gastric stomach cancer, those kinds of things, right? If someone talks about pain in their right lower quadrant, appendix would be right at the top of the list, right? Acute appendicitis, maybe colitis, ulcerative colitis, all kinds of things. So you get the idea. It's these quadrants and these regions are there to narrow down your search of a potential pathology and the body region and the organs, underlying organs that you're looking at. All right, let's answer these questions. What type of plane would separate the nose and mouth into superior and inferior structures? You tell me, superior and inferior. Transverse, remember, transverse section gives you that. Which directional term would be most appropriate in the sentence? The elbow is dash to the wrist. The elbow is proximal to the wrist, right? The elbow is closer to the attachment of the arm to the torso, to the shoulder. So the elbow is proximal to the wrist, okay? The wrist is distal to the elbow, conversely, okay? The term antebrachial refers to which body region? Forearm. Remember, go back and see forearm. Which body cavity is associated with the lungs? Pleura. Talked about that as well. What are the names of its serous membranes? Parietal serous pleura, parietal pleura, and visceral pleura. Okay, it's the same membrane. It just doubles up and folds onto itself. What is found in between these two membranes? A pleural cavity, which is filled up with a serous fluid called the pleural fluid. If a physician makes an incision into the abdomen along the mid-sagittal plane, visualize this, superior to the umbilicus, okay, uh, and just inferior to the thoracic diaphragm, it's superior, higher up to your belly button, to the umbilicus, and just inferior to the diaphragm, then the skin of the dash abdominal pelvic region has been incised. What is this region right here then? Epigastrium. So this is an epigastric incision that the physician, the medical person has just made. Okay, so what is homeostasis? We know that. Constant internal conditions, very important for life. You cannot survive if your body temperature kept fluctuating from 100 degrees to like 50 degrees or something. So core body temperature, 98.7 degrees Fahrenheit. Same with your blood sugar, blood water, all kinds of things. They have to be maintained in those narrow ranges, okay? So how does this whole thing work? It works like a machine, like a biological machine, okay? So what is the control center for your homeostasis? This organ at the base of the brain called the hypothalamus. Very fascinating organ, one of my all-time favorites, all right? It's also called the 4Fs organ, and I'll talk about what those 4Fs are later on. So this is your body's very own thermostat and hunger and thirst regulator right there, okay? So, so how does this whole thing work? Okay, so first step, you have the stimulus, and in this case, the stimulus might be uh, your temperature is getting too hot in the room, right? Then the receptor picks up this stimulus. What would be the receptor in this case? The sensory nerves in your skin. Just, you're feeling hot. And so these impulses from the receptor go to your control center. What's the control center here? The base of the brain, the hypothalamus. And then the hypothalamus will fire off signals to the effectors. What are the effectors? The organs that actually respond to you feeling hot. What organs respond to your feeling hot? Your sweat glands, they start to sweat. And what else? Your uh, blood vessels, they dilate close to your skin. So you turn this ruddy complexion, reddish tinge to the skin because you're sweating out and you're hot. And so you end up releasing heat. Okay. So this is something called negative feedback. What is negative feedback? Simple. If you've ever operated your thermostat to regulate temperature, that's negative feedback. All right. So let's say you set your room temperature at a comfortable 72 degrees Fahrenheit, right? So what happens is if your room temperature starts to get a little hotter than that, 72 degrees, then your AC comes on. And the temperature starts to drop back towards 72 degrees. But then if it gets too cold, 70 degrees, 68 degrees or something, then the AC trips and the room starts to warm up again. And then again, when it gets hotter than 72, your AC, so you are maintaining the level at that set point, 72 degrees Fahrenheit. That is exactly what happens with your body's negative feedback processes, right? Things are basically maintained at a baseline. When they go too up, too high, you bring them down, your body brings them down. When they go too low, your body brings them up. That's called negative feedback, all right? So then what is positive feedback? The exact opposite, which means that one action just strengthens the same action more in one direction, okay? So what are some examples of positive feedback? We talked about negative feedback, that is like temperature control, all of that, that makes sense. What are some examples of positive feedback? So the examples are breastfeeding, as you see here in this picture, also giving birth, labor contractions, and blood clotting. Okay, so how? What is positive feedback? Okay, look at breastfeeding here. The lady has put the baby to the breast. So as the baby suckles at the nipple, right, suckles at the breast, this sucking action sends sensory nerves, sensory signals through the nerves from the breast to the hypothalamus of the mother. The hypothalamus and the pituitary gland, which is just below the hypothalamus here, basically responds to these sensations by secreting a hormone called oxytocin. Oxytocin goes to the breast and causes the milk to be expelled out so the baby can drink it. So the more the baby drinks, the more the baby suckles, the more sensations that get to the brain, to the pituitary gland, the more oxytocin is secreted, the more milk is expressed. And this keeps happening till the baby is getting his or her fill. Okay, so this is positive feedback as far as breastfeeding is concerned. What happens with childbirth? So childbirth, when the baby is ready to be born, and the baby usually comes head first, right? So the head of the baby is stretching the walls of the cervix down there, right? The cervical walls, when they stretch, they send sensory signals back up to the pituitary gland. That's the onset of labor. The labor has started, birth pains have started. So uh, the posterior pituitary again responds to these stretch signals by secreting a hormone called oxytocin. Oxytocin is transported down to the uterus, and it makes the uterus contract. Oxytocin causes uterine smooth muscles to contract, pushing the baby's head further down, which stretches the cervix even more. The more the cervix is stretched, the more these stretch signals make their way up to the posterior pituitary. The more the posterior pituitary secretes oxytocin, the more intense the labor contractions. And so you see, this is a positive feedback, more and more and more uh, forceful, strong uterine contractions till the baby's head is past the cervix and the baby is eventually born that way. All right. Another example of positive feedback. Do keep in mind, though, that positive feedback is usually is helpful in very limited number of cases. It's usually pathological. Most of the time, negative feedback is what you need, but there are a few exceptions, like breastfeeding and childbirth and blood clotting. That's an example of positive feedback, right? Where there's a happy ending. But most other types of positive feedback are actually detrimental, such as congestive heart failure. Think of that. When your heart is failing, right, it's not pumping enough blood. So when it's unable to pump enough blood, it's also unable to pump enough blood to itself. So when not enough blood is going to the heart itself, it's getting weaker. So the weaker it gets, the lesser the amount of blood it pumps. The lesser the amount of blood it pumps, the lesser blood that goes to the heart itself. So it gets even weaker. And the weaker it gets, the lesser blood it pumps, the lesser blood it pumps, the less strong and forceful the contractions till the heart stops altogether and you die because of cardiac failure. Okay. And so that's positive feedback ending in a disruptive condition. Okay. So list and describe the three components of a homeostatic system. What were they? Hmm. What was that? Your receptor, then your control center, and then the effector, right? Those are the three. Examples of each. So your receptor could be your skin. It's picking up temperature signals. Your control center is your base of the brain, the pituitary gland. And your effectors are your sweat glands and the skin, which let go of the heat. On a cold day, what are some of the strategies the body uses to conserve heat? You shiver, of course, and then your hairs stand on end, making use of these goosebumps muscles called the erector pili muscles that traps heat closer to the skin surface as well. So what is the main difference between a homeostatic system regulated by negative feedback and one regulated by positive feedback? So we talked about the difference. Negative feedback brings the response in the opposite direction as the stimulus. Positive feedback keeps going in the same direction, okay? So here are some examples of normal ranges in order to maintain human life within the homeostatic boundaries. Body temperature average 98.6 degrees Fahrenheit. Uh, Blood glucose levels have to be clinically between 80 to 110 milligrams of deciliter per deciliter of blood. Blood pressure in between 90 to 120 upper read and 60 to 80 millimeters diastolic. Okay, uh, so basically, and now keep in mind, even though these are like numbers that apply to a vast majority of humans, 5% of healthy population have values outside these normal range. For example, personally for myself, my blood pressure ever since I remember, and I think going back to my teen years, okay, and I was quite fit, not too bad still, um, was my systolic was always in between 128 to 130 millimeters of mercury, okay? It never was 100, anywhere near 120 or less than that, all right? Over 70 to 75 millimeters of mercury for myself till my teens. All right. So, uh, so that is not the usually prescribed 120 over 80 millimeters of mercury situation, but that is not the normal for me, right? So I happen to be one of those outliers as an example. So keep that in mind as well. So what happens when this homeostasis is disrupted? You're unable to maintain this regulation, this control in the body. Let's take a look at an example. Diabetes mellitus. All right, there's two types, type 1 and type 2, which we will talk about more later on. Diabetes is a disease where you are unable to maintain your blood glucose, your sugar levels within the homeostatic range. Okay, so it's elevated higher than 110 milligrams per deciliter of blood on a regular basis. Okay, and this is a result of the ineffectiveness of your insulin production. Insulin is a protein hormone secreted by your pancreas, which regulates your blood glucose levels, right? So if the insulin is ineffective in doing its job, that's when you develop diabetes mellitus, okay? So, um, and what causes it, it's a whole separate topic of discussion that we will get into later on. So here you're looking at how clinicians use the scientific method. Again, uh, they have a question or an observation, then they come up with a hypothesis, they do a treatment or an experiment, they gather data from it, and then they basically draw up a conclusion and come to a definitive diagnosis, okay? All right, so here's an example of where certain drugs actually act by acting on your normal homeostasis. Okay, for example, patients with depression, one of the most highly prescribed, widely prescribed drugs of all time is Prozac, right? Prozac, which is an antidepressant. And so what Prozac is, uh, like many of the other drugs prescribed in psychiatry, it's an SSRI, and that stands for selective serotonin reuptake inhibitor. Serotonin is a neurotransmitter, is a chemical in the brain that regulates your mood, makes you happy. It also induces sleep, makes you drowsy, and your emotional state. So serotonin is concerned with all of that. Also bone growth, something else, okay? And so in patients with depression, the levels of serotonin are low, either genetically, okay, or because of some disease process, because of some diet that is not giving them adequate serotonin production or whatever, but the serotonin levels are low. So what you do here is you give them SSRIs, drugs that selectively block the reuptake of serotonin in the brain, so the levels of serotonin rise. When the levels of serotonin rise, these patients kind of come out of their depression, all right? And but since again, too much serotonin can cause troubles too, as you can see here, right? Digestive becomes hyperexcitable. You might develop diarrhea, nausea and vomiting, upset stomach, also bone growth, bones become weak. So you have to get it in the right range, all right? So just one example of what can go wrong with homeostasis. So here are some examples of medical imaging. So you take a medical history, remember, then your medical exam, which is four things, again, inspection, palpation, percussion, and auscultation. And then you come up with a tentative diagnosis, but how do you get a definitive diagnosis? You send for blood work, lab work, and you do medical imaging like what you see here. So what are some examples? A radiograph, which is a simple X-ray. X-rays are penetrating electromagnetic radiation that give you a picture of the uh, uh, hard tissue of the body, like teeth and bones, as you can see here. Remember, a mammogram in all women 40 years and above are encouraged to get the mammograms at least once every 3 years to seek any unusual lumps early on. Uh, that's a mammogram is nothing more than a plain X-ray. That's all it is, right? And it shows any solid mass like a calcified tumor or something which might have formed in the breast. So radiograph, obviously looking for bone and teeth pathologies as well for hard tissue. Then here you're looking at a CT scan or computed tomography. This again utilizes X-rays, but here you can also visualize the soft tissue, as you can see the brain here. And CT scans are widely used for things like stroke detection and tumors and whatnot. All right, so that is a CT scan that you see here. Then you have something called an MRI, which stands for magnetic resonance imaging, and no X-rays or radiation is used here. In fact, you're put into this tunnel, the MRI tunnel, right? And you're bombarded with highly energized magnetic waves, and they cause the protons in your body to line up in a certain way, and then they take a picture of that, and that gives this image right here. All right, so the benefits of MRI, some of them are there's no radiation used, so there's no danger of radiation poisoning, cancer, whatever, right? Uh, and it gives you a rather detailed picture of the underlying soft and hard tissue as well. So then you have the ultrasound or sonograph, and that's widely used in like to look at an embryo or a fetus, right? So during prenatal checkups, uh, doctor's going to use a sonogram or an ultrasound to look at the baby. And this is completely harmless. These are high frequency sound waves, nothing to do with radiation. You can also use an ultrasound to look for like renal stones, stones in your kidneys or gallstones in the gallbladder, right? Also blood flow in your carotid artery. You can use an ultrasound, basically a Doppler ultrasound to look at how good that blood flow is to the brain in cases of stroke and stuff, right? All of those applications. Then what you're looking at here is something called digital subtraction angiography. What happens here is you inject a dye, a radio opaque dye, a dye which shines or glows in the dark, if you will. And this dye spreads throughout your arteries. And wherever the dye does not go or fails to go, and that area is not glowing, it's dark, that tells you where the blockage is. So if you've injected this person with a radioactive dye, and you don't see any coloration here, it stops here. That tells you there's some kind of an obstruction, either a tumor or a stone or something, a cholesterol plaque, which is blocking the blood here. All right. Finally, you have something called a PET scan or a positron emission tomography, which is usually done in the brain. And what the PET scan does is it picks up the metabolic activity of an organ, right? So the areas which are glowing red, bright red here, these are the most metabolically active. They're getting the most blood, they're using up the most oxygen and nutrients. Uh, and then come the blue areas, the next most active, and then the greeners, the dark green, and then the light green ones are the least active areas. And I can tell you a lot about this person just by looking at the PET scan here. I can tell you that this person is intensely looking at something, looking visually. Secondly, whatever he or she is looking at has activated his some memories in him. All right. And this person is also trying to make some connections between what he's looking at and his emotions and the memories that this image is eliciting in this person. So how in the world was I able to tell you all that just based on a bunch of colors in the brain? So that's where you have to know the brain structure, the anatomy, and the physiology of the brain. All right. So let me quickly take notes here. PET scan and what the person is feeling in the picture. Okay, so first up, what I see is what is the most metabolically active area um, in this picture, right? So here, the occipital lobe, the back of the brain. The occipital lobe is where you process visual information, what you see through your eyes. So that's the most metabolically active, glowing bright red. He's obviously, or she is obviously looking at something. So how did I know about memories and emotions? Look at the temporal lobes. You see those two bright red dots there? So that's the memory and the emotional areas of the brain. So whatever he or she is looking at is eliciting some emotions there, right? And how did I know that this person is trying to make sense of what those emotions are, putting them into a into context? Look at this bright red spot in the right frontal cortex. This is the area, the frontal cortex, which basically makes sense of the memories and puts them in the proper perspective, in the proper context. So just based on that knowledge, you can see that what's going on in this person's brain. So the PET scan can also be used to pick up areas like cancer. So cancers will glow bright red because cancer, what is a cancer? Mutated cells in your body which are starving their neighboring cells of all the oxygen and food because they're stealing all of that and growing and multiplying and refusing to stop, right? So I call them the psychopathic cells of the human body. That's what they are. They act like psychopaths, right? They have no remorse. They have no concern for the surrounding cells. They charade and masquerade as normal cells. They're very good at evading detection, and yet they're malignantly growing and invading your body. So the psychopaths of the human cells, right? And so since they take an inordinately higher amount of oxygen and nutrients from the surrounding tissue, they tend to be metabolically more active. And so they tend to glow bright red in a PET scan. So you can also localize and uh, focus on certain cancers and know where to do surgery, where to do chemotherapy, radiotherapy to get rid of that. All right. So what is an example of a disease process by which homeostasis is disrupted? You can tell me now. Diabetes mellitus, right? A failure of regulation of your blood sugar levels, right? That's one example. So, or depression, where your serotonin levels are too low. Okay, so there we are. We are done with our chapter 1 that was on the schedule for today. So um, again, remember to keep up with the labs. I already sent out the instructions for that. Okay, and again, do not stress over getting everything turned in and perfectly technically correct the first time around. Do as much as you can and turn it in, and I will grade it. Let me know if you have any questions there. All right. Keep up with the recorded lectures and the notes that I'm going to send out with this. Shortly, I will be sending out the study guide for your first lecture exam, which is next Tuesday, a week from now. So hopefully you'll have the long weekend to go over that information. You have my email address. Any questions, suggestions, feedback, interesting articles to share, whatever, please feel free to do that. And then so enjoy the rest of your evening. Have a wonderful session. Take care, stay safe, and I will see you all virtually, that is, hopefully tomorrow in the evening. Bye.